Translation is the second stage in gene expression, where a nucleotide sequence in DNA or its RNA transcript is converted to amino acid sequence in a protein. Three types of RNA molecules perform different but complementary roles in protein synthesis. mRNA carry information copied from DNA in the form of codons. tRNA deciphers the code and delivers a specific amino acid. rRNA associates with set of proteins to form ribosomes that finally make the protein. In this entire process, a key topic is centered around genetic code. Genetic code describes relationship between nucleotide sequence in DNA or its RNA transcript and sequence of amino acids in a protein. For this lecture, we will cover 4 topics. The first topic is about mRNA and genetic code. Second topic is about tRNA. Third topic is about rRNA. For the fourth topic, we will examine steps of translation. Now, let's go to part 1. Genetic information is arranged in codons. Each codon is made up of 3 nucleotides corresponding to 1 amino acid. Genetic information is derived from DNA. After transcription and splicing, the genetic information is carried by mRNA in the form of codons to be translated into a protein. Genetic code describes the full set of relationship between codon and amino acids. It is typically presented in a table like this. Each codon is written from 5' to 3'. When you look for a codon, you look for the first nucleotide on the left column, and the second nucleotide on the top row, and the third nucleotide on the right column. For example, when you look for the amino acid that UUC codes for, you look for the first U on the left column, second U on the top row, and C on the right column. You will find here UUC. It codes for phenylalanine. 3 nucleotides, each having 4 choices. The possible combinations are 64, but we have only 20 amino acids. Therefore, there must exist several codons that code for the same amino acid. Indeed, except methionine and tryptophan, all other amino acids have more than 1 codon. In addition, there are 3 stop codons, signal for end of translation. The genetic code has several characteristics. First, nearly universal. The same genetic code is used in all organisms with minor exceptions. Second, specific. A codon codes for a specific amino acid. For example, GCC always codes for alanine. Third, redundant. As I described to you earlier, more than 1 codon can specify a single amino acid, except methionine and tryptophan. Fourth, non-overlapping. The ribosome reads continuously 3 bases at a time. For example, this is the first codon, followed by second codon, third codon, and fourth codon, continuously, non-overlapping. Fifth, commaless. The codons are read continuously, no break, no comma. 6. No direct interaction with the amino acid. Instead, the tRNA serves as an adapter that bridge codon and amino acid. Any mRNA sequence have 3 possible open reading frames. For example, this is an mRNA sequence. It has 3 possible open reading frames. These possibilities remain valid till the start codon is specified. Once the start codon is set, all the remaining codons are determined. Besides codons which are contained in coding region, eukaryotic mRNA also have 5' untranslated region or 5' UTR and 3' UTR that I described to you before. These sequences are also important. For example, 5' UTR and 3' UTR may have impact on nuclear export, may serve as the binding sites for regulatory RNAs, may bind regulatory proteins to increase or decrease translation. 5' UTR may contain internal ribosome entry sites, which are often used by RNA virus as a means to ensure viral translation is active when host translation is inhibited. 5' UTR may also contain upstream operating frames, which typically inhibits translation of the downstream primary operating frame. 3' UTR may contain AU-rich elements that the regulatory proteins bind to, which typically increase mRNA stability. Now let's move to part 2. Primary structure of tRNA features unusual bases such as dihydrouridine, pseudouridine, ribothymidine, and inosine. tRNA carries amino acid at the 3' end, right here. The secondary structure of tRNA has 3 features. First feature, tRNA has 4 main arms: D-arm, T-arm, acceptor arm, and anticodon arm. Some tRNA also have an extra arm or variable arm. Each arm typically has 1 loop and 1 stem. The second feature is intermolecular base pairing occurs in 4 stems. Third feature, 4 arms form clover-leafed shape. The tertiary structure of tRNA features L-shaped 3D structure, such as this L-shaped, that allow them to fit into the P and A sites of the ribosome. Here I highlighted 2 arms. First, anticodon arm that reads a codon, and acceptor arm that carry an amino acid. Aminoacyl tRNA synthetase are the enzymes that charge the tRNA. One amino acid tRNA synthetase for each amino acid, with a total of 20 such enzymes. Some amino acids have several codons. As a result, these amino acids have several different isoacceptor tRNAs. Since only one amino acid tRNA synthetase for each amino acid, all isoacceptor tRNA for a particular amino acid use the same enzyme. Substrates for aminoacyl tRNA synthetase are amino acid, isoacceptor tRNA, and ATP. A special initiator tRNA begins translation. It's carrying methionine in eukaryotes and formylmethionine in prokaryotes. This is how it works for tRNA charging. First, amino acid is loaded onto adenosine monophosphate and become activated. Second, the amino acid is then transferred to the 3' end of tRNA. Now this tRNA is charged. Each anticodon contains 3 bases in the tRNA anticodon loop. 3 bases of the anticodon complementary to the codon on mRNA. tRNA are named according to the amino acid they carry. For example, this is phenylalanine tRNA, proline tRNA. There are 61 codons for 20 amino acids. Each amino acid is carried by at least one tRNA. If each codon had its own tRNA to match with, it would require 61 tRNAs. In fact, we have only 50 tRNAs in the cell. It means some codons will not have a corresponding tRNA. This is a challenge. Solution to this challenge: one tRNA can recognize more than one codon. For example, alanine has 4 codons. These codons only differed at the third base. If one alanine tRNA can recognize all these 4 codons, we will only need one tRNA instead of 4 for alanine. This would require base pairing between anticodon and codon not to be strict. This leads to Warburg hypothesis. Warburg hypothesis states usual pairing occurs at the first and second bases of the codon. Here is mRNA, and this is a codon. Usual pairing occurs at the first and second base of the codon. Unusual pairing occurs between the third base of the codon and the first base of the anticodon. For example, U can pair with G, and vice versa. G can pair with U. Unique to tRNA that has inosine, inosine can pair with U, C, or A from codon. With the Warburg hypothesis, one tRNA can recognize more than one codon. Among codons for one amino acid, first two nucleotides have the greatest similarity. Third nucleotide has the greatest variation. For example, glycine has 4 codons. The first two nucleotides are the same. Variation occurs on the third nucleotide. Rule of Warburg applies to the third position of the codon. At this position, U can pair with G from the anticodon, and vice versa, G can pair with U. And A, U, or C from the codon can pair with inosine from the anticodon. Here is an example of normal pairing, and here is an example of Warburg pairing. This is a summary of unusual base pairing between codon and anticodon. Warburg base of anticodon is not strict in base pairing with codon. As a result, one tRNA recognizes multiple codons. This is a summary of base pairing between anticodon and codon. Unusual base pairing is highlighted in blue. Now let's move to part 3. In prokaryotes, the large and small ribosomal subunits are 50S and 30S respectively, which combine to make 70S ribosome. In eukaryotes, the large and small subunits are 60S and 40S respectively, which join to make 80S ribosome. As you may notice, prokaryotic ribosome and its subunits contains odd number 3, 5, 7. In eukaryotes, they contain even number 4, 6, 8. This feature can help us distinguish between these two types of ribosomes. Ribosomes contain 3 discrete sites. Amino acid site, or A site, is the first binding site for tRNA. It binds new coming tRNA molecule carrying an activated amino acid. Peptidyl site, or P site, is the second binding site for tRNA. It holds the tRNA which is linked to the growing polypeptide chains. It also binds initiator tRNA. Axis site, or E site, is the third and final binding site for tRNA in the ribosome before it exits. Now, let's move to part 4. Very similar to transcription, translation also goes through initiation, elongation, termination stages. Next, let's look at each stage in more detail. We will start from prokaryotes. In prokaryotes, 30S ribosomal subunits bind Shine-Dalgalo sequence upstream of AUG, the start codon. With the help of initiation factors, 50S subunit and initiator tRNA join. The initiation complex formed. This is described here. Initiation of translation in prokaryotes require mRNA, initiator tRNA, and ribosomal subunits, and initiation factors. The initiator tRNA recognizes the start codon. It carries a methionine that is covalently modified to N-formyl methionine. This is N-formyl methionine. The start codon is AUG in most cases, but in some cases, GUG and UUG are also used. All codes for N-formyl methionine. The Shine-Dalgalo sequence is a ribosomal binding site in bacteria and archaeal mRNA, generally located around 8 base upstream of the start codon AUG. The 6th base consensus sequence is AGG-AGG. In prokaryotes, initiation can occur at internal AUG with a Shine-Dalgalo sequence. Here is an example. Lac-operon in E. coli is comprised of LAC-Z, LAC-Y, and LAC-A genes. In regions covering LAC-Z and LAC-Y, there are 3 AUG codons, 2 with Shine-Dalgalo sequence and 1 without. Initiation of translation occurs at the 2 start codon accompanied by Shine-Dalgalo sequence, but not the one without. In eukaryotes, initiation can only occur at first AUG downstream of 5' cap. Internal methionine codon cannot serve as an initiation site. The assembly of initiation complex in eukaryotes is very similar to that in prokaryotes. Major difference is more factors are needed, especially more initiation factors. In eukaryotes, initiation factors are denoted EIF. E refers to eukaryotic. Another minor difference, initiator tRNA that carries methionine instead of formyl methionine. Initiation starts when a number of EIFs bind the 5' cap of messenger RNA. More detail is shown next. The eukaryotic initiation factor 2 binds GDP and initiator tRNA and delivered initiator tRNA to the 40S subunit. The small subunit then binds the 5' cap and scans down the mRNA to find the first AUG codon. The initiation codon is recognized, and GTP is hydrolyzed to GDP. And EIF2 dissociates from the complex. EIF2 will be recycled. Recycle of EIF2 is subject to control by phosphorylation, and this will be discussed in lecture B11. The large ribosomal subunits then bind the small subunits and tRNA and place the methionine tRNA in the P site. This is the P site and this is the A site. Initiation is now complete. In most cases, eukaryotic 40S ribosomal subunits locate the start codon by binding to 5' cap and scanning down to find the first AUG. But around 5 to 10% of cases, ribosomal subunits bypass first AUG, scanning for more favorable one, which is defined by Kozak's rules. If A of AUG is set as +1 position, according to Kozak's rule, a favorable context should have purine at -3 position and G at +4 position. Here is the example. This is mRNA, and this is +1, and this is -3, and this is +4. So the ribosome will enter at the 5' end of mRNA and scan down the RNA to look for the first AUG that is in good context. A favorable context occurs when purine at -3 and G at +4 positions. In eukaryotes, the elongation factor EEF1 binds GTP, an amino acid tRNA, and delivers the tRNA to the A site. Once it arrives in the A site, GTP is hydrolyzed to GDP, and elongation factor 1 is released from the complex. In prokaryotes, elongation factors are EF1 and EF2. Formation of peptide bond occurs when the amino group from the A site attack the carbonyl carbons in the P site. This is a nucleophilic addition reaction. At the end, the polypeptide is transferred from the P site to the A site, and this is the result. This reaction is catalyzed by peptidyl-transferase. This enzyme is contained within a sequence of 23 sRNA in the prokaryotic large ribosomal subunit, corresponding to 28 sRNA in eukaryotes. The energy for peptide bond formation comes from ATP used for tRNA charging. Following peptide bond formation, elongation factor 2 catalyzes the GDP-dependent ribosomal translocation step. The newly formed A site-bound peptidyl tRNA and P site-bound empty tRNA move to the P site and E site respectively. Shift 1 position. Now, the ribosome is ready for the next tRNA. The energy for elongation is provided by 1 ATP and 2 GTPs. 1 ATP is used for tRNA charging, 1 GTP for elongation factor 1, and another GTP for elongation factor 2. Here is clinical correlation. Diphtheria toxin is an exotoxin produced by Corynebacterium diphtheriae, a gram-positive bacteria. Diphtheria toxin can inactivate elongation factor 2 through ADP ribosylation, leading to diphtheria. Symptoms of diphtheria include low-grade fever, chills, fatigue, cyanosis, sore throat, and blood-stained nasal discharge. Initial diagnosis is based on symptoms. Diphtheria should be suspected when the patient presents with upper respiratory tract illness with sore throat, lower grade fever, and great pseudomembrane covering the posterior of the pharynx. This is the example of the pseudomembrane. A similar toxin, exotoxin A, is produced by Pseudomonas aeruginosa, which is a gram-negative bacteria. Pseudomonas aeruginosa is a type of multi-drug resistant pathogen. This pathogen typically caused ventilator-associated pneumonia and various sepsis syndromes. Exotoxin A inactivates elongation factor 2 through ADP ribosylation in a manner similar to diphtheria toxin. When translation reaches the stop codon, a release factor binds within the A site, recognizes the stop codon. Release factor catalyzes hydrolysis of the complete polypeptide from the peptidyl tRNA, and the entire complex dissociates. The components of this complex are recycled. The three-dimensional structure of release factors is very similar to that of tRNA. Eukaryotes have only one release factor. Bacteria have three of them. A feature associated with translation is polysome. Several ribosomes binding to one mRNA and translating the message at the same time. This occurs in both prokaryotes and eukaryotes since mRNA is typically much longer than ribosome. Here is integration with immunology. When cells are infected by virus, the infected cells produce interferons. Interferon bind the receptor on the neighboring normal cells. Normal cells synthesize antiviral proteins. These cells are now protected by these antiviral proteins. Here are examples of two antiviral proteins. One is 2,5-A synthetase. This enzyme synthesizes oligo-2,5-adenylate. This is a compound in which nucleotides are joined together through 2,5-phosphodiester bond instead of 3' to 5' phosphodiester bond seen in nucleic acid. This oligo activates endonuclease, which cleaves viral mRNA. Second protein is eIF2 kinase. This enzyme phosphorylates eIF2. Upon phosphorylation, eIF2 is kept in inactive state. Translation initiation is blocked. Here is integration with pharmacology. A group of antibiotics block translation at various steps. These antibiotics can be summarized in one mnemonic, ATCCM. It represents American Type Culture Collection in Manassas, Virginia. This is a very popular cell collection center. Here, A represents aminoglycosides, including streptomycin. These antibiotics binds to the 16S rRNA of the small subunit and blocks loading of initiator tRNA to T sites. So these antibiotics block initiation step. All the other antibiotics in this group block elongation step. Tetracycline blocks binding of the amino acid tRNA to the A site. Chlorphenicol block peptidyl transferase activity. Clindamycin and macrolides, including erythromycin, block translocation step. of translation.